This is chapter 5 of your textbook. It's talking about the structure and function of large biological molecules. So things that are really important in living things. This is actually 2 parts, so this is part 1 of this chapter. So these are what we call macromolecules. These are large molecules that are really essential for life. Carbohydrates, which break down into sugars. Proteins, which break down into amino acids. And nucleic acids, which are your DNA and RNA, so they break down into the components like nitrogen bases. They break down into phosphate groups. They break down into nucleotides, which the 3 parts of the nucleotide are the base, the phosphate group, and then the deoxyribose sugars. Lipids are technically not large enough to be a true macromolecule, but important enough that we should discuss them as well. Macromolecules may also be called polymers. A polymer is just a long molecule made of many similar or identical building blocks. So we kind of go back to it and think, these are the monomers. Sugars are the monomers of carbohydrates. Amino acids are the monomers of proteins. And these 3 things make up a nucleotide, which is the monomer of a nucleic acid. And then lipids are made out of different things depending on what it is. So one of the ones we'll talk about is a phospholipid, and that's made out of like a lipid tail and a phosphate head. So it just kind of depends on what the lipid is, exactly what it's comprised of. Let's see. Monomers, those are your building blocks. Synthesis and breakdown of polymers. So building or breaking polymers occurs with the help of enzymes. Okay, remember we've already talked about these enzymes speed up a chemical reaction. They are an example of a protein, and they are an example of how shape is really important to function. Because if the shape of the enzyme is not correct, it's not going to perform its function. For example, people that either lack the lactase enzyme or the lactase enzyme is warped or not correctly shaped, they will not be able to break down milk sugars. And we have more on enzymes later where we do a lab with them. 2 important reactions. Dehydration reactions is when you're building a polymer. So you're taking monomers and you're making polymers. That doesn't look very good, but that's what I'm saying. Hydrolysis is when you're breaking down a polymer into its simpler parts of the monomers. How are these reactions different? Well, they're different in how they use water as part of their reaction. That's one of the ways that they're different. And we're going to show that here in a little bit. Go back to the word hydrolysis. Think about anything with hydro means water, and then dehydration. Well, if you're dehydrated, you're lacking water. So you'll see that water even is in part of the name of that reaction. So here's a dehydration reaction. This is when we're synthesizing or making a polymer. Hang on one moment. Okay. So in this case, we are actually going to remove water from the equation. So here is a short polymer, and we're going to link that with another monomer. So each time we're adding a monomer, say this is a protein, and we have the amino acids over here. Each time we add an amino acid to the chain, we're going to build or make or remove a water molecule to make that happen. So the hydrogen in one part is going to link up with the hydroxide functional group in the other, and that's going to separate and bring out a water. That's why we call it a dehydration. We're actually removing water. Think about your cells or yourself. If you're dehydrating, you're removing water. What you get then is a long polymer. Now, if you wanted to add another amino acid, you could add it right here, where the H group linked up with the OH group on the other one, or you could add it here, where the hydrogen group linked up with the OH group on that. Hydrolysis is when we're breaking down a polymer. So we're taking a polymer, something large, and we're breaking it down. So again, say we're going to start with a protein here. We're going to break it down into its simple amino acids. Now, in a dehydration reaction, we're producing a protein when we're starting with the amino acids. So here, we want to break this apart right here. Okay, we're going to break it down. So we actually have to add a water to that reaction. Okay, hydrolysis adds the water. Think about like hydroplaning with your vehicle. You're adding water to the road, and that's causing you to lose control. So hydrolysis adds the water. And that's going to break this bond right here. And the hydroxide group is going to go with one amino acid. And then the H group is going to go with the other one. Remember, in a dehydration reaction, we're simply putting those two things together. In a dehydration reaction, in a hydrolysis reaction, we are separating that. And so each end, one end is going to get the hydroxide group and hydroxyl group, and one end is going to get the hydrogen. And we're adding the water in there to make that happen. Dehydration, remember, again, is used to build polymers from monomers. It creates a water in the process. Example is during the formation of proteins from simple amino acids. You're going from amino acids to a protein here. And that shows you that picture again of the dehydration. And then hydrolysis is disassembling the monomers, where the bond between the monomers is broken with the addition of a water. So the hydrogen from the water attaches to one monomer, and the hydroxyl group from the other one attaches to the other monomer. An example is digestion, when complex polymers, so things you're eating, are broken into smaller monomers to be absorbed into the bloodstream. So for example, when you eat a bagel is made out of a lot of carbs, carbohydrates. Those carbohydrates go through hydrolysis reactions to break that bagel down into sugars, simple sugars like glucose. And that's important because diabetics, this is kind of linking up a big idea here, when that blood sugar rises in glucose, that could be a problem if individuals don't have insulin, because insulin helps to take that glucose into the cells. And if people don't have insulin, then they can't regulate that glucose level in their blood. So they have to limit the number of carbohydrates they eat. If you notice that about diabetics, they can't eat a lot of carbohydrates because of the sugars that they break down into. So here, we're talking about the hydrolysis reaction. So let's talk a little bit about some of these different macromolecules. Carbohydrates are what we call complex or just regular carbohydrates, but they're all made out of simple sugars, which are called monosaccharides. Disaccharides are double sugars, so a little bit more complex. Polysaccharides are multiple sugars linked together. So most carbohydrates are going to be some kind of a polysaccharide. Major source of fuel for the cell. Why? Because it's going to break it down into glucose. And glucose is what we need for cellular respiration. And cellular respiration produces ATP. It's an energy source that your cells use. That's why carbohydrates are a really good fuel for the cell, because they're producing ATP. Some polysaccharides serve as storage material. For example, starch in plants or glycogen in animals, or it can even be building material for the structure of the cells. So cellulose and chitin, and those are things that chitin is something like I'm looking for what I'm looking for. Insects that have hard shells have a lot of chitin in them. That's always important, and that's a type of polysaccharide. Cellulose is something that you might find in like plants that animals can't digest. Interestingly, cows have a rumen that easily digests cellulose. Stored fuel cannot sustain an animal for long, and they're depleted within about a day, and they need to be replenished. So you can't just not eat. You have to replenish those carbohydrates in order to continue to produce ATP through cellular respiration. This is actually an issue with low carbohydrate diets. It works because your body is not going to produce as much glucose, and glucose will, when not used, when you're not active, glucose will turn into fat. It'll store it as fat. But if you're on a low carbohydrate diet, you might not get enough carbohydrates to actually make enough ATP in order to have a lot of energy. So people with low carb diets can actually be kind of sluggish. Cellulose is grouped uniquely with few organisms. All right, sorry, I've been getting pesky phone calls from solicitors on my cell phone recently. All right, cellulose is grouped uniquely, and few organisms have enzymes that can break that down. Again, cows are an example that can. Bless me, sorry. Keton is used in arthropods. That's what the word I was looking for. Insects that have exoskeletons. Those are the words I was looking for. Some fungus actually has keton as a component of them too. So here are some pictures of carbohydrates, some simple sugars. You can find carbohydrates in ring form, or you can also find them in linear form. And this shows the dehydration reaction in a synthesis called maltose. So remember that we're taking, we're going from a dehydration reaction we're synthesizing, right? So we're going from small monomers to big polymers. And so we have to remove a water from the process. So two glucose molecules will be linked together, and that will remove a water, and that will produce a disaccharide called maltose, or a more complex sugar. A dehydration reaction with the synthesis of sucrose. So you're taking a glucose and a fructose, but they're both simple sugars. You're removing a water from the equation, because that's what dehydration reactions do. And you're producing something called sucrose, which is a disaccharide, because it has two simple sugars combined together. This shows some pictures of some carbohydrates: amylose, amylopectin, glycogen. These are some microscopic pictures, as well as what the actual molecular structure looks like, although they're very simple simple drawings. Here is cellulose fibers. You can see them under a microscope. You can see them here. This is the cell wall, is made out of a lot of cellulose. So here's a more a closer picture of that cell wall. And then you can actually see they're actually made out of these fibers. And what are these fibers? These are long groups of carbohydrates. They're made out of simple sugars. And then here is a picture of keton on the exoskeleton. And this is the structural formula for that. It's actually also used to make strong, flexible surgical thread because it decomposes after the wound or incision is healed, because it's actually a biological molecule. All right, so lipids, we said, are not really true polymers, but they're really important to talk about. So the major function here is energy storage. Okay, lipids are also known as, okay, think about that. People that have a little extra fat, they're storing all of that energy that they weren't using. Lipids are grouped together because they mix poorly with water, and so that's what makes it something a lipid, is that it doesn't mix with water. Fats are really the same thing as lipids. Sometimes they separate them into different categories. For example, triglycerol and glycerides are actually fat, whereas a phospholipid is not really a fat, but it is an important component of cell membranes. It's a lipid. Fats contain a glycerol and a fatty acid. These are their monomers, the simple parts. Saturated fat is straight with no double bonds. Most animal fats, they are solid at room temperature because of that. Omega-3 fatty acids are actually essential and need to be supplied in the diet. You can get this a lot from fish. Unsaturated fats contain a double bond and that actually bends the structure. It provides a kink in it. Most of these are plant and fish fats. They are liquid at room temperature. The bend means they can't back closely enough to actually solidify at room temperature. These are actually the better ones for you when you're talking in terms of fats. Obviously, none of them are great. But the unsaturated fats are better for you than the saturated. So, for example, this would be vegetable oil or olive oil. This would be bacon fat, lard. Trans fats have a special double bond, and they're actually the worst. So you'll see a lot of products that say no trans fats, and they're really highlighting this because in the media we've shown, or we try to educate the public that trans fats are bad. These are actually man-made fats that have a special double bond. They're not good for you at all. They really contribute to heart disease. And it's really funny because sometimes you get something that says no trans fats on it. You have things like carrots or something, and you're like, really? But they really are highlighting that because people see that and they're like, oh wow, I need that because that's good for me. So here is a fatty acid and a glycerol that's making up a fat. Notice here you have a dehydration reaction. Here is a triglycerol. Notice it's tri, meaning 3. And so you've got the 3 glycerols here and the 3 fatty acid chains. Saturated fats, remember I said that they were straight, okay? So they can pack really closely with each other. You have one here and one here, and they can pack together, and because of that, they become solid at room temperature. Unsaturated fats don't. They have this bond here, this double bond that kinks it, and so they don't pack as tightly like these. They have to be a little bit more loosely packed. And so because of that, they don't solidify at room temperature. They're a liquid. Phospholipids are important because they make up the cell membrane. This also shows how well the form fits its function. Remember, the cell membrane is what's going to kind of help let things in and out of a cell. And it will work in regulating what comes in and out based on its structure. A phospholipid membrane is made up of a lipid tail and a phosphate head, and these lipid tails are actually tucked into the center of the membrane. So if this is the membrane going all the way around, they're tucked into the center, and so they actually stay away from the water environment because remember, lipids don't dissolve well in water. So that makes it important for its function. 2 parts, heads and tails. The tails are hydrophilic, meaning they are okay with the water. That was this part up here. And then the tails are hydrophobic, which means they are phobia. They have a fear of the water. And that's this part right here with the tail. The cell membrane is designed so that the hydrophobic part is away from the environment and the hydrophilic portion is in contact with the environment. We'll talk more about this when we get to the cell membrane. Steroids, lipids with 4 fused rings. Cholesterol is actually a steroid. It's part of the cell membrane, but it can also, if you have too much of it, can contribute to heart disease because it is a lipid. So steroids are lipids. And then hormones are actually lipids, and this is where a lot of people don't realize is that they are lipids. Hormones like estrogen and testosterone and all those that we secrete, there's obviously more than those growth hormones. Anything that is a hormone in your body is in fact a lipid or a steroid. So this shows the phospholipid. Okay, this is kind of how we normally draw it. This is a little bit more accurate. This shows the bonding to it. So this is the fatty acid tail. Remember, this part is phobia. This is hydrophobic. It doesn't like to be around water. And this part is the head that is hydrophilic. And it's okay with the water. And then this is a picture of that membrane. So these are the hydrophilic heads, and these are the hydrophobic tails. And notice that the hydrophobic is in the center because they fear that water. So the only part that's actually exposed to the extracellular fluid, which is composed of a lot of water, is the part that is okay with that. And then this is a picture of a cholesterol or a steroid. because steroid is a cholesterol, or cholesterol is a steroid. Okay, and that ends the chapter 5, part 1 notes. I'll get to part 2 here soon.